In the previous video, we looked at diffusion, osmosis, and tonicity. These are three concepts that you should be familiar with from chemistry and cell bio. If they are unfamiliar to you or confusing, you should do a little bit of background reading in our textbook or in Module 0. So now we're looking at roots, because roots are all about water for plants. So root hairs are unicellular projections. They're just one long cell from the epidermis, that outer layer, of growing root tips. And all next week we'll be looking at root and shoot anatomy. So this is something that you should hopefully see this week when your fast plant seeds germinate. Fast plants are related to mustards, so if you are familiar with like mustards that have seeds in them, that's about the size of your fast plant seed. It's a tiny, tiny, tiny little reddish seed. So you are going to sprout those. You're going to germinate them. And the first things that will come out will be the first two leaves. So those are your cotyledons. That's what you see up here. And a radicle. And the radicle generally emerges first. So the radicle is this early root system. And notice that it's covered in this fuzz. Each of these little strands is a root hair. Root hairs are specially adapted. Their whole purpose is for water absorption. So if we look at these under the microscope, we'd see these long unicellular projections, no cross walls in there. They have high surface area and low volume. And we know that that is to maximize interaction with the external environment. Their whole job is absorbing water from that external environment. So this is really the interface where plants are going to get their water from, is from these growing root tips covered in root hairs. As roots age, they, in many plants, will get a outer layer that is dead called bark, and those are no longer absorbing water. So all of this water absorption that happens in a root really happens at the growing root tips that are out there scavenging. They can also do it through their mycorrhizal symbionts, right, who are out there in the soil scavenging water and nutrients. But it's all going to happen from these growing tips and then from those mycorrhizal partners. Where am I out of the way? I'm just going to move my face. The water is pulled into the plant through the root hairs. So it's going to do that because the plant is going to contain higher solutes than the external soil environment. So water is going to want to move into the plant through these root hairs. And it diffuses into the vascular cylinder of the root. So there has to be a bit of a concentration gradient here for us to get the water from the soil into the root hairs and then into the vascular cylinder. It needs to want to move that way. Otherwise, we don't want to take energy to have to push it or transport it. So we need to have a concentration gradient, a relatively high concentration of solutes in any of these cells that are around the xylem cells, and then lower concentration of solutes in the external soil environment. And you'll see all of this anatomy next week when we look at root anatomy. Okay, so water molecules flow into the xylem, which forms a conduit of pipes. These, sclerenchyma cells, they're not sclerenchyma. These lignified cells are dead at maturity, allowing water to flow through the empty lumen of the cells. So you have seen these in your celery cross-sections and maybe in some of your other plant tissue samples. These here, these long cells we're looking at, if we were to take a plant growing this way, we would slice it longwise and look inside of it. That's what we're doing right now. It's a long section. And we're looking at these long tubes from the side, as opposed to straight on as you're doing in your thin cross sections. So here we're looking at these vessel elements from the side, and we can see their secondary walls are all that's left, and they have these spiral thickenings. And depending on when the xylem was formed during the plant's growth, what type of plant it is, it might have different patterns for that secondary wall. The ones that are formed first have to stretch really far, and so often they'll just be made of rings. The ones that are a little bit later might be spiraled, and then ones that are formed even later might be more of this, I think it's called scleriform, where they're just full of these bordered pits everywhere. So all of these are xylem cells, and they have that thickened secondary wall, and it's lignified, so it's just like a woody pipe that water is going to travel through. So now water needs to move up the plant. Roots are located down in the soil or whatever the substrate is, whatever they're attached to. We need to transport the water up to those photosynthesizing parts that are going to use that water for photosynthesis. How do we get the water there? We're working against gravity. So the water is pulled upward against gravity by attractive forces because water is amazing. Water's polar. It has positive and negative charges on it, partial charges. So not only will water stick to itself, but water will also stick to other surfaces that are also polar. So here it's showing cohesion, unfortunately, between the two oxygen molecules, or atoms, because it's the oxygens and the hydrogens that stick together. So there's negative charges on the oxygens. Oxygens are super negative, and hydrogens are super positive. So they're attracted to each other. So cohesion is this mutual attraction. It's a similar attraction, so water being attracted to water. Even though it's opposite charges on that water, it's the same molecule. So for same. Adhesion, think of adhesives that stick together, right? Adhesion, where the term adhesive comes from, is water sticking to non-water surfaces. So these are both going to be important components of how water is going to travel upward. So that water gets pulled upward against the forces of gravity, and it's going to use capillary action, or that's how we're going to sort of describe this whole process. So capillary action has to do with the ratio of cohesion to adhesion. For water, the cohesive forces, water sticking to itself, aren't as strong as the adhesive forces of it sticking to the sides of that container. So if we have a higher volume and a lower surface area for it to stick to, like in a wider tube, relative to the amount of water that's in there, there's less surface of the tube for it to stick to. We have more cohesion, and that's not as strong, so it's not as much adhesion pulling it upward. We get more cohesion that's causing the water to stick together, but not enough adhesion to work against gravity. If we have a skinny tube, that's quite a lot of surface area of the tube relative to the amount of water that's not touching that surface. So we have more adhesion relative to cohesion, and the adhesive forces are stronger, so the water starts moving up that tube against the forces of gravity, trying to stick to more of those surfaces. So the water gets pulled up through a thinner tube. The thinner that tube gets, the more adhesion to cohesion ratio there is, and so the higher it can climb against the forces of gravity. Here's a little demo of that, or kind of where those forces are. The adhesive forces are just here on the end. Cohesive forces are here in the middle. So we need more adhesion to be able to pull up these water molecules. And that's going to be different for different molecules, like mercury in a thermometer. The mercury actually has stronger cohesive forces, and so it will pull itself downward. I think the thinner the tube is. The wider the tube, you might be able to get it to pull itself upward. But so it has a sort of a opposing capillary action to water. Okay. So veins of xylem and phloem branch off from the stem and into the leaves. So we're going through the xylem here, from the roots up into the stem and then out into the leaves. So here we have a skeletal leaf. This would be the midrib here in the center. We're looking at a eudicot leaf, and it has this netted pattern of venation. You can see with all of that leaf tissue gone where all of the veins are, and it's an incredible network, right? And it's compartmentalized into all of these little like vein neighborhoods. And a little fun fact, no cell is more than 6 cells away from a vascular bundle here. So as leaves decompose, often the xylem is the last to break down because of its lignin content, and that can leave these really cool skeletal remains. I find this particularly obvious in holly leaves and tomatillo shells. They're the two things that I find around the most that have this really cool, like, cagey skeleton that you can look at. And if you were to add toluidine blue to this, it would turn that nice bright aqua blue because it's that secondary cell wall structure. Okay, I'll pause here since we're at 10 minutes and continue in the next slide. So we've just tracked this movement of the water from the roots, or from the soil, into the roots through the root hairs, into the xylem, up through the stem, and out to the leaves. So we're going to start with the water in the leaves and what it's doing.